Hi, my name is Mia Temkin, and the title of my project is The Effect of Capsaicin on the Mitochondrial Viability of Breast Cancer Cells. The purpose of this experiment was to identify how the mitochondria in cancer cells react to capsaicin. Specifically, if capsaicin affected the overall efficiency or metabolic activity of the cancer cells. I decided to test the hypothesis that if you introduce breast cancer cells to varying concentrations of capsaicin, then the mitochondrial viability will be decreased. Capsaicin is the chemical in hot peppers that makes them spicy. The chart you see is a basic comparison of the intensity of capsaicin in various hot peppers. In this experiment, I worked with pure capsaicin. Capsaicin causes apoptosis in some cancers, which is why it has cancer-prohibiting effects. Apoptosis is programmed cell death. Essentially, a chemical reaction occurs beginning in the mitochondria that tells the cell that it can no longer function, so it commits suicide so as not to damage the body. Capsaicin does not affect healthy cells. The reason it affects cancer is this. While normal cells perform anaerobic or aerobic respiration, cancer does both at the same time. This means that while normal cells are producing about 32 molecules of ATP per one glucose molecule, cancer produces 44 molecules of ATP per one glucose molecule. This is called the Warburg effect, and it allows the cancer cells to metastasize and survive in places they couldn't otherwise survive. It also allows them to leach nutrients from other cells. The basic difference between cancer and healthy cells, metabolically speaking, is illustrated in this picture. As you can see, the cancer cell takes a lot more nutrients from its environment than the healthy cell does. The cancer cell does this by overproducing an enzyme called hexokinase 2. Hexokinase 2 is a glucose magnet. It attaches to glucose and acts as a transporter to the cell. In the picture, the DNA is blue and the hexokinase 2 is green. Here are my results. I measured the mitochondrial viability with a stain every 24 hours and used a spectrometer to get the absorbance of that stain at 570 nanometers of light. What I found is that over time, increasing amounts of capsaicin correlated to a decreasing amount of mitochondrial viability. Essentially, as the concentration of capsaicin increased, the cells became less metabolically active. I used statistical analysis to verify these results. I used a two-variable t-test. Whenever I compared the highest and lowest concentrations of capsaicin to the control, the t-test showed that my results would almost always happen. However, when I compared the concentrations between each other, the t-test showed that these results were more likely to happen by chance. I took this to mean that capsaicin would decrease the mitochondrial viability of cells. However, a higher dose may not mean more effective treatment. My results have effectively proven my hypothesis. This experimentation could lead to a further understanding of the metabolism of cancer. Understanding the metabolism could lead to further targeted cancer treatments. When you deprive an organism of energy, it can no longer function. If one were to use the metabolism of cancer as a target for cancer treatment to deprive it of energy, it would create a more effective and efficient cancer treatment. Capsaicin could also be used as a treatment that doesn't harm healthy cells, which is different than anything used today. Today we have things like chemotherapy and radiation, which actually harm healthy cells. Using capsaicin as something that targets only the cancer, you would eliminate that battle against the side effects of the cancer drugs. Also, this research could lead to a better understanding of how cancer reacts to harsh chemicals, whether or not it treats cancer, which could help doctors and researchers predict how the cancer will react to certain treatments.